Hi friends, welcome to UNS classes. We are back with another previous year question from biology. This time question is from PCR, polymerase chain reaction. So here the question is, which is the correct order of PCR or polymerase chain reaction? So as usual, you know, first we will discuss something about PCR, come to know the process, and we will answer the question, right? So let's go to what is a PCR. So what is PCR? Polymerase chain reaction. For what it has been done? This is done to amplify a small fragment of DNA, whether it may be a small piece of DNA from a crime scene or from fossils or anything. This process will help us to amplify the amount of DNA so that we have so many copies, or we can say that this is an alternative of cloning, right? So let's check what are the process involved in PCR. So in PCR, the components that we need are, definitely we need DNA sample, right? We need a DNA sample, then we need a primer, right? In DNA replication, we know what and all we need. We need a DNA template, a primer, DNA polymerase, ligase and all. Similarly, here we are gonna replicate the piece of small DNA that we have got. So we need a primer, definitely we need nucleotides. What are the nucleotides we will use here? Since it's DNA, adenine, thymine, guanine and cytosine, right? We need nucleotides and we need Tac polymerase. Tac polymerase is nothing but a DNA polymerase, right, to synthesize the DNA strand, and we need a buffer, mixed buffer and a PCR tube to put in this PCR machine, right? Now let's check the different stages of PCR. What are the different stages? Normally it has 3 stages. First one is denaturation, annealing and elongation. So what is denaturation? While studying DNA replication, for that what we do unwind the DNA, right? Unwinding DNA is nothing but breaking the hydrogen bond between 2 double strands. That's called denaturation. It's called denaturation because here we apply a huge temperature to break the hydrogen bonds between 2 strands of a DNA. So here the optimal temperature that we use is around 94 to 98 degrees Celsius. Between 94 and 98 degrees Celsius we use the temperature to denature the DNA strands. See, the hydrogen bonds between these DNAs will be broken and we'll get 2 independent strands of DNA as templates, right? Now if we write, it will be like 5' 3' direction. Here also we are using 5' 3' direction. But normally how a DNA will be, it will be like 5' 3' direction and 3' 5' direction, right? For the easy representation, we are making both the strands like this. Now the next step is annealing. So while annealing, see the temperatures has been reduced to 50 to 68 degrees Celsius, right? Normally the optimal temperature will be around 64 degrees Celsius. Okay, so the temperature has reduced. So during the process of annealing, we reduce the temperature from around 94 degrees Celsius to 64 degrees Celsius. Here what we will do? Here we need a, what is this? We have seen it here, right? We need a primer. Primer is an RNA. We need a primer. Primer, right? Now what's the function of primer? To just initiate the process of replication or an extra strand of DNA which is complementary to the template, right? The next step is elongation. Elongation is nothing but elongate the add new nucleotide to the template, what template we have here. For that we need DNA polymerase, but if you see the temperature of elongation is 72 degrees Celsius. So the normal DNA polymerase present in human body, will it work in 72 degrees Celsius? No, right, because the normal human body temperature will be 36 degrees Celsius in which only these enzymes will function, or the enzymes need the optimal temperature for the functioning. So at such a high temperature we can't use normal DNA polymerase. For that we have Tac polymerase. Tac polymerase is nothing but the polymerase which are taken from the bacteria who lives at very high temperature, or we can say that they are thermophilous in nature. So the bacteria which are thermophilous in nature, thermophilous, and the bacteria from which we are extracting Tac polymerase is called Thermus aquaticus. thermus aquaticus, right? So by using this enzyme we will do the elongation process and we will get an extra copy. So we began with a one DNA, one double stranded DNA. Now in the end we got two double stranded DNA, right? This cycle will repeat to a point where we want the cloned too many numbers of DNA. How much number we want, we will repeat this cycle, right? So what are the steps in PCR or polymerase chain reaction? We have denaturation, annealing and elongation, right? Now let's go back to the question. Here the question is, which is the correct order of PCR, right? Now if you check the options, say first option is, so first let us write the proper order. What is the first order? What was denaturation, right? Denaturation is nothing but breaking the hydrogen bonds between two double stranded DNA, and the second step was annealing. Annealing is nothing but hybridization, right? Hybridization between DNA and RNA, DNA strand and the RNA primer, right? And the third step is elongation, or we can call it as extension. Elongation or extension. Now we know the order: denaturation, annealing and elongation. So let's check which is the option here. Here first there are only two options which starts with denaturation, annealing, extension. So option B is the answer, right? Now before stopping, let's go to some more 2-3 questions which are related to PCR. See, here the question is, during the process of gene amplification using PCR in a very high temperature is not maintained in the beginning, then which of the following PCR will be affected first? So what is the question? If we don't maintain high temperature in the beginning during the process of PCR, which is the first step which will be affected? Just now we have seen the different steps. First step was denaturation, which requires around 94 degrees Celsius to break the hydrogen bond between two double stranded DNA. So what will be the answer here? Here the options are ligation, annealing, extension and denaturation. Obviously the answer will be denaturation, right? Here the answer will be denaturation. Okay, now the next question here we have is, okay, which of the following is not an application of PCR or polymerase chain reaction? Check the options. What are the options here? Detection of gene mutation, molecular diagnosis, third one is gene amplification, and the fourth one is purification of isolated protein. See, we know what is PCR. PCR is nothing but amplification of a small fragment of DNA. So what is DNA? What does DNA contain? DNA contain genes, right? Genes or the codons, right? So what is the first option here? Detection of gene mutation. See, DNA contains genes. By amplifying a gene or amplifying a DNA, we can understand different gene and we can understand whether there is any gene mutation, right? So it's an application of PCR, right? What is the second option is molecular diagnosis. Molecular diagnosis is the same, that is in a molecular level, that is in DNA level. We will check whether there is some mutation or any other changes in a DNA. So it is also an application of PCR, right? See, all these are in DNA level only, it's going on. Now the third option is gene amplification. Obviously we told that what is the function of PCR, or why do we need PCR? To amplify the DNA. What's DNA? DNA is a gene, right? Gene is a fragment of DNA. So obviously this is also a part of PCR, right? Now the last option is purification and isolation of protein. See, protein is the second or the third stage of DNA replication, right? First the DNA has to replicate. From that replicated DNA, we will create some mRNA, and from the mRNA, we produce protein using ribosomes, right? So in a PCR, we are only using DNA and DNA amplification. So there is no protein in that. In all other three options are in DNA level. The fourth option is protein. So what will be the answer? Which is not an application of PCR here is purification and isolation of the protein. Hope by discussing all three questions you got the concept of PCR properly. Happy learning. Bye. See you in next class.